discuss the placenta. Now differentiation of the two layers of the trophoblast. So what is happening? The cells near the decidua, they lose their cell boundaries and become one continuous sheet of cytoplasm called as syncytium. The layer of trophoblast is called as syncytiotrophoblast or plasmodiotrophoblast. Now deep to the syncytium, the cells of the trophoblast retain their cell wall and form a second layer called as cytotrophoblast or Langerhans layer. Now next step is syncytiotrophoblast grows rapidly and become very thick. Now small cavities they called as lacunae appear in this layer, you can see, which gradually enlarges. Now at first they are irregularly arranged and later on they are arranged radially around the blastocyst. Now this is the initial part, that is the syncytium is there, syncytiotrophoblast you can say. In these the small cavities they appear, later on they fuse to form the bigger one. Now initially these small cavities they are arranged randomly, but later on they are arranged radially around the blastocyst. Now lacunae are separated by partition of the syncytium called as trabeculae now. So this is the part of the syncytium which forms the trabeculae. Then lacunae gradually communicate with each other and form one large space. Each trabeculus is now surrounded all around by this lacunar space, you can see. So these are the lacunar spaces and these are the parts of the syncytium which now they are called as trabeculae. Syncytiotrophoblast grows into the endometrium. So this is the decidua basalis side. So syncytiotrophoblast they grow towards that. As the endometrium is eroded, some of its blood vessels are opened up and blood from them they fill the lacunar spaces. Now each trabeculus is initially made up of entirely of syncytiotrophoblast. Later on the cytotrophoblast begins to multiply and they grow into each trabeculus. Now each trabeculus has central core of the cytotrophoblast surrounded by the syncytiotrophoblast, which is further surrounded by the maternal blood which is filling the lacunar spaces. Now trabeculus is now called as primary villus and lacunar spaces is called as intervillus space. Now it has a core of cytotrophoblast surrounded by syncytiotrophoblast. So this is primary villus. Now extra embryonic mesoderm. On this side the extra embryonic mesoderm is there lining the inner side of the trophoblast. Now invade the center of each primary villus. So this is how it is happening. So the villus thus comes to have a core of mesoderm, outer to which is cytotrophoblast, which is further surrounded by syncytiotrophoblast or syncytium. Now this is known as secondary villus. Now secondary villus has the central core of extraembryonic mesoderm surrounded by cytotrophoblast, surrounded by syncytiotrophoblast, and outer to this in intervillus space the maternal blood is present. Now soon thereafter the blood vessels they enter into, which are lying in the extraembryonic mesoderm, they enter the mesoderm of the villi, forming the core of each villus. Now villus is fully formed and is called as tertiary villus. So tertiary villus has capillaries, then there is the extraembryonic mesoderm, then outer to the cytotrophoblast, then syncytiotrophoblast. So this is tertiary villus. Fetal blood now circulates through the villi, and while maternal blood circulates in the intervillus space. Now cytotrophoblast that grows into trabeculae that does not penetrate the entire thickness of the syncytium, therefore does not come in contact with the decidua. The cells of the cytotrophoblast now spread out to form a layer that completely cuts off the syncytium from the decidua. Now this layer is called as cytotrophoblastic shell. The cells of this shell multiply and placenta increases in size. So the villi formed are attached on the fetal side to the embryonic mesoderm. On the maternal side it is attached to the cytotrophoblastic shell. Now these villi are called as anchoring villi. Now further, if we discuss the subdivision of the villi, the anchoring villi give off numerous branches which grow into the intervillus space. You can see, so this is the anchoring villi. This is the anchoring villi. Then anchoring villi includes the stem villi or truncus cori. So it has the anchoring villi has a trunk. Then stem villi branch into rami cori. So this is rami cori. Then further is the subdivision of this villus is ramuli cori. So this is ramuli cori. Now anchoring villi branch and rebranch, you can see in this diagram, to form the free or floating villi. So filling the intervillous space, increasing the surface area which is available for exchange between maternal and fetal circulation. Because in intervillous space the maternal blood is lying, and in the villus the fetal blood capillaries they are present containing the fetal blood. New villi are sprout out from the chorionic side of the intervillous space. So ultimately, the whole of the intervillous space becomes filled with villi, increasing the surface area available for exchange between maternal and fetal circulation. Newly formed villi at first consist of, as we have discussed, in sensitio trophoblast, then subsequently invaded by cytotrophoblast, then mesoderm, then blood vessels. So forming the primary, secondary, and tertiary villi respectively. Now placenta becomes subdivided into number of lobes by septa growing into the intervillous space from the maternal side. You can see these septi. Such lobes are called as maternal cotyledon. The number is around 15 to 20. If the placenta is seen from the maternal side, so if it is seen from the maternal side, the bases of the septi are seen as grooves and cotyledon appear as a convex mass. Each lobe contains number of anchoring villi and their branches. So each maternal cotyledon has number of anchoring villi and their branches. One such villus with its branches is called as fetal cotyledon. If one such villus is present, so this is known as the fetal cotyledon. And if the number of villi which are separated by the different septi coming from the maternal side, so they form one maternal cotyledon. Now coming to the structure of placenta, it has three parts: maternal side or basal plate, you can see. So this is the maternal side. Then fetal side or chorionic plate. So this is the fetal side. Then between basal plate and chorionic plate, so this region. Now maternal side basal plate is made up of decidua basalis, cytotrophoblast shell, syncytiotrophoblast, and fetal side is made up of amnion, extraembryonic mesoderm, cytotrophoblast shell, and syncytiotrophoblast. And between these two plates, the intervillous space is present, so in which the maternal blood passing through the intervillous space, fetal blood flowing through the fetal villi, stem villi, and its branches. If we see the full term placenta, diameter is around 6 to 8 inches and weight is approximately 500 grams. After the birth of the child, the placenta is shed off along with the decidua. Now if we see the maternal surface is rough and is subdivided into cotyledons. So the septi they appear as grooves, and the raised area or cotyledon they appear as a convex area. Now this is the structure of placenta. Now coming to the fetal surface is lined by amnion. It is smooth and is not divided by into the cotyledon. The umbilical cord is attached to this part, that is on this surface the umbilical cord is attached. Coming to the placental membranes, in the placenta the maternal blood circulates through the intervillous space and fetal blood through the blood vessels in the villi. Now maternal and fetal blood do not mix with each other. All interchanges of the oxygen, nutrients and waste products take place through this membrane. So that is the placental membrane. Now what are the structure forming this membrane? So endothelium of the fetal blood vessel. We know it that in the core the fetal blood vessels they are lying, then surrounding mesoderm, then cytotrophoblast and its basement membrane, then syncytiotrophoblast. So these structures they form the placental membrane. If we see the structure of one villus, so from center to periphery, so these structures are present. Towards term, disappearance of the cytotrophoblast layer occurs from the most of the villi, you can see. So the efficiency of the membrane is increased. So it become highly permeable. Now the placental membrane has capillary endothelium and a very thin layer of syncytiotrophoblast that separates the fetal from the maternal blood. Now towards the end of pregnancy, efficiency is reduced because of the fibrinoid deposit on the surface of chorionic villi. Coming to the syncytial knots, it consists of aggregation of the nuclei in the syncytiotrophoblast. These knots break off and enter the maternal circulation, may be lodged in the capillaries of the maternal lung where they finally disappear. Coming to the functions of placenta, it enables the transport of oxygen, water, electrolyte and nutrition from maternal to fetal blood, provides excretion of carbon dioxide, urea and other waste product from fetus into the maternal blood, act as a barrier and prevent bacteria and other harmful substances from reaching to the fetus. However, most viruses, some bacteria, and drugs taken by the mother can enter the fetal circulation. While permitting the exchange of several substances between maternal and fetal blood, it keeps these blood streams separate, thereby preventing antigenic reaction between them. Then it synthesizes progesterone, estrogen, and some other hormones. Progesterone secreted by placenta is essential for maintenance of pregnancy after degeneration of the corpus luteum. Now coming to the umbilical cord. Umbilical cord is a cord-like structure connecting the placenta and the fetus. Length is about 50 centimeter. Structure is it contains two umbilical arteries, right and left, and one umbilical vein, that is left one, which are twisted around each other. Now vessels are embedded in a mucoid connective tissue called as Wharton's jelly. So in this Wharton jelly is there. Now it is covered by the amnion then remains of the allantois they are present, then remains of the vitello-intestinal duct, that is seen. So these are the content of the umbilical cord. As the embryo folds, expanding amnion surround the embryo. Amnion also encloses the connecting stalk, vitelline duct, yolk sac, and allantois. These structure constitute the umbilical cord. Normally, vitelline duct, yolk sac, and allantois degenerates, and the umbilical cord is left with umbilical vessels and Wharton's jelly. Now, functions of the umbilical cord. Umbilical arteries arising from the ventral division of internal iliac artery carry deoxygenated blood from fetus to the placenta. After oxygenation in the placenta, the umbilical veins carry oxygenated blood from placenta to the fetus. It is to remember that placenta is an organ of respiration for fetus. Coming to the anomalies of placenta. So considering the shape, it may be bidiscoidal, two discs may be formed, it may be lobed, divided into various lobes, it may be diffuse when chorionic villi present all around the blastocyst, as we have discussed in the previous lecture, that is chorionic levi and chorionic frondosum. Normally towards that end, that is towards the decidua capsularis, the villi they disappear, but if they persist, we call it as diffuse placenta. Now placenta succenturata, small part of the placenta is separated from the rest of it. Then fenestrated or hole in the disc may there. Then circumvallate placenta, peripheral margin is covered by circular fold of decidua. Now variation in the attachment of the umbilical cord to the placenta. Normally this umbilical cord is attached in the center, but if it is attached to the margin, we call it as battledore placenta. You can see here, this is normal, this is marginal. Then furcate placenta when blood vessels divide before reaching the placenta. Then filamentous placenta can be there when blood vessels are attached to the amnion where they ramify before reaching to the placenta. Now normal sites of the implantation of the ovum. Attachment of the placenta may extend partially or completely into the lower uterine segment, and we call it as placenta previa. Now placenta previa has various degrees. In first degree, attachment of the placenta extends into the lower uterine segment. So normal position of the placenta is in the upper uterine segment. But if it is lying in the lower uterine segment, we call it as placenta previa. This is the first degree, which is in which the placenta is extending up to into the lower uterine segment. In second degree, the margin of the placenta reaches the internal os but does not cover it. Third degree is the edge of the placenta cover the internal os, but when the dilatation of this os is happening, so the placenta does not occlude the internal os. In fourth degree, the placenta is completely covering the internal os and it occludes even after it was dilated. So this is all about today's lecture. So this lecture is very, very important. You may get question in the form of write a note on placenta, write a note on the formation of the placenta, then what are the different congenital anomalies of the placenta. So these are all the important questions which you can expect from this topic. Thank you. Thank you very much.